Hi everyone, I hope you're all safe and well. My name is Dan Santos and I'm a human geographer, and I will be presenting my seminar in October. So my seminar will largely draw upon some of the findings from my dissertation research, which examined the democratization of biotechnology in community science labs. And to provide some brief context and background to that, over the last 10 years or so, biotechnological materials and tools and resources like DNA sequencing and more recently CRISPR-Cas9 have become much more widely available and accessible to a larger range of publics. And as a consequence, community science labs have become established in various places around the world. And what these labs do is provide the resources and the space for people to conduct their own experiments and to pursue their own projects outside of mainstream scientific institutions in academia and industry. And the sorts of people who come to these labs and who are part of these communities are pretty diverse. They include interested amateurs, engineers of various kinds, high school and university students, and professional working scientists. And in the last few years or so, members of these community science labs have gotten together and held conferences at MIT, an important research institution for synthetic biology, and declared themselves a movement. And one of the aims of this movement is to democratize biotechnology. And so what I tried to do in my dissertation and what I'd like to do in the seminar is to sort of unpack what democratizing biotechnology means in practice, right? So if community science labs are now a global phenomenon with broad overall aims, how are these aims translated and implemented in particular labs located in particular locations, right? So how do geographical dynamics at various scales, from the local scale to the regional to the national scale, affect the meanings and the possibilities and limits of democratizing biotechnology in practice? And so to do this, I focus on two community science labs, two case studies, both of which are located in the San Francisco Bay Area, a globally significant hub for biotech. And what I think is really interesting about these two labs, one of which is called Counterculture Labs, located on the northern end of the Bay in Oakland, and the other called BioCurious, located in the southern end of the Bay in Silicon Valley, is that despite the fact that both of these labs are sort of, share similarities with respect to attitudes towards technology, given their location in a sort of globally significant region for technology, each of them arguably has distinct visions and imaginaries and understandings around what the democratization of biotechnology should ultimately be for. And so what I'll do in my seminar is sort of provide some ethnographic insights and evidence as to how these two communities differ and how the knowledge and practices, both scientific on the one hand and social on the other, inform and undergird what democratizing biotechnology means for these particular communities. And ultimately, I'd like to argue that if biotechnology is being democratized on a global scale, then remaining attentive to the geographical differences and dynamics is important going forward. So again, thank you to the BEC for providing me with this opportunity to share some of the findings of my research, and I hope to see you in October in the seminar. Thank you.